Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CUET Common Entrance, that is Central University's Common Entrance Test question paper for the subject Biochemistry, which was conducted in the year 7th June 2023. This is part 3 of the explanation. If you are new to my channel, you can like, share and subscribe. If you want still previous year question papers of biochemistry, you can refer them in my channel playlist. I have uploaded them there. Okay, first question. Malate, fumarate and succinate precede the formation of oxaloacetate in the citric acid cycle. Find the option which shows correct order of the formation in citric acid cycle. So this is the citric acid cycle. They have given malate, fumarate, succinate. You have to find the order until oxaloacetate. So here if you see the cycle, it is first will be succinate, then fumarate, then malate and then oxaloacetate. So the answer here will be D, B, A and C. Answer is option 1. Then next one. Phosphoryl transfer reaction are essential for the energy content of a cell. Find the correct answer in which the following phosphorylated molecules have been arranged in a decreasing order of their phosphoryl transfer potential. So the least ones will be glucose 6-phosphate and glucose 3-phosphate. Then adenosine triphosphate will be between 1,3-bisphosphoglycerate and phosphoenol pyruvate. So the first one will be phosphoenol pyruvate. It is more in phosphoryl transfer potential. So C will be first, then comes 1,3-bisphosphoglycerate, B, then comes ATP, adenosine triphosphate, then D, that is glucose 6-phosphate and then glucose 3-phosphate. Answer is option 1. Then next one. In analogy with central dogma of biology, how will you arrange the following omics in a correct sequence? Metabolomics, proteomics, genomics, transcriptomics, phenotype or phenomics. So first it will be genomics. Why? Because DNA is getting replicated to DNA. From DNA you create RNA, that is transcriptomics. From RNA you get it converted to proteins, so it is proteomics. From proteins to metabolomics, then from metabolomics it will be the phenotype or the phenomics. So the answer here will be C, D, B, A, E. Answer is option 3. Then next one. Nucleotide, nucleoside, purine and pyrimidine are the building blocks of DNA and RNA. Based on their molecular weight, arrange these molecules in an ascending order. So first one will be your phosphate, E. Then comes your pyrimidine. Pyrimidine is a single ring compound and purine is a double ring compound. So pyrimidine will come next and then it will be purine. So CECA, then nucleoside and then nucleotide, B. So the answer here is option 4, ECADB. Then next one. Reverse transcriptase is used for synthesizing cDNA from RNA. Which of the following options represent the correct order of the given steps? Annealing of the oligo-DT primer to mRNA, DNA polymerase 1 and DNTPs form double-stranded DNA. Reverse transcriptase form complementary single-stranded DNA. mRNA is degraded with alkali. Downstream analysis of double-stranded DNA. So first one will be annealing of the oligo-DT primer to mRNA, A. Then comes after annealing of the oligo-DT primer to mRNA, then you use the reverse transcriptase to form cDNA. Then you get an RNA DNA hybrid, which you have to degrade the mRNA. So mRNA is degraded with alkali. Then you take DNA polymerase 1 and DNTPs to form double-stranded DNA from cDNA. Then downstream analysis of the double-stranded DNA. So the answer here is option 1, A, C, D, B, E. Then next one. DNA cloning is an essential tool in recombinant DNA technology. Arrange the different steps of DNA cloning in proper sequential order. Restriction digestion of insert and plasmid, selection of host cell or transformants having recombinant DNA, in vitro DNA ligation, transform. Yes, in vitro DNA ligation. No, first will be restriction digestion of the insert and plasmid, A. Then you go for in vitro DNA ligation. Then after ligation, you go for transforming it into a suitable host cell. So transformation of the ligated DNA. Then you select the host cells for recombinance. So B will be selection of host cells or transformants having the recombinant DNA. Then confirmation of the recombinant DNA by sequencing. E. So answer here will be option 3, A, C, D, B, E. Then next one. Hydrophobic nature of amino acids affect the tertiary structure of protein. Which of the following option shows the given amino acid in decreasing order of their hydrophobic character? So the least hydrophobic is lysine. Histidine is is hydrophilic. So lysine is the least. So last will be E, lysine, and before that will be histidine. Then the others we will see. So most hydrophobic will be phenylalanine. So B. Then comes leucine, A. Then comes glycine, then histidine, and then lysine. So the answer here will be option 2. Then next one, two statements, statement 1 and 2. Statement 1. Cooperativity in receptor ligand interaction causes large change in receptor activation with small change in ligand concentration. Yes, that's correct. In amplification, the enzyme associated with signal receptor is activated, which in turn activates many downstream enzymes in a so-called enzyme cascade. That is also correct. So both statements 1 and 2 are correct. Answer is option 1. Then next one. Statement 1. Phototrophs and chemotrophs derive their energy from sunlight and chemical fuels respectively. That is correct. Phototrophs and chemotrophs cannot be autotrophs but only heterotrophs. That is not correct. Because phototrophs, they are light energy users. There are two types, that is autotrophic phototrophs and heterotrophic phototrophs. So autotrophic phototrophs, they use light energy and fix carbon dioxide into organic compounds, that is plants and cyanobacteria. Whereas heterotrophic phototrophs, they also use light energy but obtain carbon from other organic sources, that is purple bacteria. And then you have got chemotrophs. They are also two types, that is autotrophic chemotrophs and heterotrophic chemotrophs. Autotrophic chemotrophs, they obtain energy from chemicals to fix nitrogen into organic compounds, that is sulfur oxidizing bacteria. Then heterotrophic chemotrophs, they also use chemical energy but obtain carbon from other organic sources. So the answer here is statement 1 is true but statement 2 is false. Answer is option 3. Then next one. In naturally occurring 20 amino acid, both carboxyl and amino group are bonded to the alpha carbon. Yes, that is correct. Statement 1 is true. According to organic chemistry IUPAC nomenclature, alpha carbon is also the C1 carbon of the amino acid. No, it is not C1. See here, this is the structure of an amino acid. This is your C alpha carbon atom. It is bound to NH2 and COH. So this is the first carbon atom, the COOH. Then comes C alpha, then your R group. So COH will be the carbon 1, not C alpha. So the statement 2 is not correct. So your answer here is statement 1 is true but statement 2 is false. Answer is option 3. Then next one. The peptide bond C alpha CN C alpha present in protein is rigid and planar. Yes. Proteins are highly dynamic and undergo large conformational changes. That is also correct. So both statements 1 and 2 are true. Answer is option 1. Then next one. Major histocompatibility complex is present on the cell surface of T cells for antigen recognition. That itself is not correct. Because T cells, they do not display MHC molecules. Instead, they recognize antigen presented by MHC molecules present on antigen presenting cells. So T cells, they do not have MHC being displayed on them. Then statement 2, during organ transplantation, the major histocompatibility complex is involved in allograft rejection. That is also correct. So statement 1 is false but statement 2 is true. So the answer is option 4. Then next one. Chymotrypsin hydrolyzes peptide bond through formation of a transient covalent acyl enzyme intermediate. That's correct. This type of mechanism in enzyme catalysis is operative in all enzymes in the biology. That is not correct because different enzymes use different mechanisms to obtain catalysis. That is lysozyme, they use acid-base catalysis. Carbonic anhydrase use metal ion catalysis, that is zinc is involved there as the metal. Then lactate dehydrogenase, they use redox reactions. So how can be that this type of mechanism is operative in all enzymes in the biology? That is not possible. So statement 2 is incorrect but statement 1 is correct. So the answer here is option 3. Statement 1 is true but statement 2 is false. Then next one. Sanger's chain termination method is used for sequencing of single-stranded DNA by using nucleotides and short DNA stretches. That's correct. Sanger's protein sequencing method uses 1-fluoro-2,4-dinitrobenzene, dancyl chloride and nucleosides for protein sequencing. Sanger's protein sequencing method is nothing but your Edman degradation. It uses phenyl isothiocyanate or the Edman reagent, not the ones which have been mentioned in the statement 2. So statement 2 is not correct. Statement 1 is correct. 1-fluoro-2,4-dinitrobenzene, it is used for amino acid identification, not sequencing. Dancyl chloride is used for amino acid analysis and protein quantitation. Then nucleosides are not used in protein sequencing. It is related to nucleic acid sequencing. So the statement 1 is true but statement 2 is false. So the answer here is option 3. What is true about bacterial DNA dependent RNA polymerase? It is an oligomer of approximately 400 kDa. Yes, that's correct. Its beta subunit together with Mg2 positive converts ribonucleotides into RNA. That is also correct. So A and B are correct. Okay, C. Alpha amanitin strongly inhibits the enzyme. Alpha amanitin is a potent inhibitor of eukaryotic RNA polymerase, not bacterial polymerase. The enzyme can also form DNA from RNA. DNA dependent RNA polymerase is an enzyme that synthesizes RNA from DNA, not the other way around. So D is also not correct. So the correct options are option 3, that is A and B only. Then next one, for the respiration, it is printing mistake. It is not respiration. It should be preparation. Of two proteins with same molecular weight, which of the following techniques could be used? Okay, they have the same molecular weight, so SDS page cannot be used. Two-dimensional SDS page can be used. One run in one direction, second one run in the second direction. So B can be used. Mass spectrometry with liquid chromatography can also be used. So gel filtration cannot be used. They have same molecular weight. Gel filtration and SDS page also cannot be used. So B and C only. Answer is option 4.